Okay, so let's make a start here then. Right, so we'll start with an introduction, and then we'll go through the whole process, starting with guiding principles, guidelines, and regulations, and then we'll work all the way through. And at the very end, we have a case study where we can show how we've applied this methodology, this process, and then we'll finish up with a conclusion. Okay, so we thought we'd start with a couple of poll questions. So the first question is here. If I could just read the question here. So which one of the following modalities would typically be considered the lowest risk in terms of potential source of viral and microbial cross-contamination? So the first one is a process that contains viral vectors or human cell lines, where an adventitious human virus will replicate. The second one is a simple MABS process. And the third one is an autologous cell therapy process, such as a CAR T process. Okay, so if you could select your question, the answer now. Okay, so the lowest risk we have 65% is the MABs. Okay, and that's the correct answer, so that's good. Okay, so that's a good start. Thank you. And then we have a second poll question here. So the question is the same, but this time with the same answers. But this time, what we'd like to know is which one do you think is the highest risk of the three? So viral vectors, human cell lines, traditional MABs, or the autologous cell therapy. Okay, we should hopefully we'll have the answers soon. Okay, so that's interesting. So we have 72%, and I can understand why. I think that viral vectors, human cell lines, it would be the highest risk, but in actual fact, it would be the autologous cell therapy process. So we'll talk, hopefully, as we go through the presentation, it'll become clearer as to why that is the case. All right, but that's useful. All right, so let's continue here then. All right, so just starting off with an introduction. So we know that ATMP or cell and gene therapy facilities are different to a lot of the conventional pharma facilities, often requiring heightened segregation. And we also know that more and more companies are having to look at how to accommodate different modalities. So some, for example, may acquire a new building, some may partially fit out part of the building. Sometimes they may not know what will go into the fallow space, sometimes they do, but we're seeing more and more facilities containing multiple modalities. And so this is becoming, of interest, I think, to more and more people within the industry, and we saw that during the COVID-19 pandemic, where it was particularly relevant when launching new vaccines. So what Tom and I did about 3 years ago is we conducted a similar study, a study along these lines, and we developed a process that worked quite well for us, and we've applied it a number of times since. So we start with the business need, we identify the different modalities, the scale of manufacture, we then gather a lot of data that then helps us develop the risk profile. We then perform a series of risk assessments, which then enable us to develop a segregation strategy. Okay. So if we start, okay. So if we start with the guiding principles, So we have an example here of some guiding principles. So, one might be, when we design new facilities, we want to design them, build them to, using the current best industry practices that are available. So this might be, closed systems. It may contain isolators, maybe single-use technology. And whilst, we work very hard to correctly categorize a particular modality, it's understood that those particular modalities may move into a higher or lower category as more information becomes available. And the intent might be that we want to benchmark industry best practices and incorporate those within the exercise. And as with all facilities, there are often constraints, operational constraints, infrastructure constraints that are important to understand that need to be factored into the study as well. So they need to be taken into account. And one of the things that, may be important as well is to think about some of the modalities that may not be suitable for a facility, that, may not be suited alongside other modalities, and to highlight those. So the guidelines and regulations are quite important. They provide quite a lot of information in terms of, what we can and cannot do. So it's important to list what the regulations, which regulations apply. So in this case, I've provided an example of some European regulations and some US FDA regulations that might apply. But it's important to list all of those and then extract information that would help us understand what modalities we can accommodate in the same facility, and provide some information in terms of how we might do that. So, for example, if we look at dedicated facilities based on the guidelines and the regulations, then highly sensitizing materials such as penicillins and cephalosporins, are examples of where we are likely to need a dedicated facility just based on the regulations alone. Where it may not be quite so clear might be with certain steroids or cytotoxic anti-cancer agents. So we would need to do the work to help understand whether we could accommodate those or not. Live vaccines would be another example where it may or may not be possible to accommodate them. And then infectious viral vectors would need to look at those very carefully as well to help understand, whether they could be accommodated or not. So the guidelines and regulations, whilst they might not give us all the answers, they're certainly giving us some useful information which would help us conduct the study. So benchmark industry best practices, we just listed 3 possible ones here that may be relevant. So the use of pass-through boxes for materials, samples, and waste to facilitate unidirectional flow and avoid mix-ups and cross-contamination. That might be a good practice that we want to incorporate. VHP decontamination may be another one. If we have human source materials, donations, and we have a site operation that contains multiple modalities, it might be that that the receipt and delivery of those human source materials is kept separate from the main warehouse area operation. Okay. Okay, and at this point, I'm going to hand over to Tom, who's going to walk through the risk profile.
Okay, just to summarize a couple of key points that Al made there. So thanks, Al, firstly, for teeing this up. So let's just take a few examples. You're a CMO and you have multiple manufacturing suites, and you want to know what modalities could be beside each other and to maximize that manufacturing space. Another example is you are a monoclonal antibody manufacturer, and you have a fallow space that could be used for a different modality, and you want to know, can you do that? Okay, or what's stopping you putting a different modality beside you? So, what we need to do is that we need to come up with a risk-based approach that actually puts a scientific argument to why certain modalities can be, possibly should not be, or definitely not beside each other. So Alpha's given out opening points there that, the regulations, they're quite definite on some things, they're a bit grey on other things, and then they say absolutely nothing about other things. So the regulations gives you some part, and then we have industry best practice that, you know, below the regulations, what are other people doing, what is best in class in the industry, and then sometimes we just have constraints that we just can't simply put different modalities beside each other. I'm going to explain that in a second. So there's two things that escalate risk when we think about mixed modality or multimodality manufacturing. One is chemical potency and toxicity. So there may be a chemical occupational hygiene hazard that escalates as we go up through a modality. So that is a purely chemical OEL exposure limit to chemical substances that might drive different approach. The second then is biological risks. So we may have that different modalities have potentially viruses present, potentially very harmful viruses present, and that risk of what's in the manufacturing process from a viral and microbial risk perspective also has to be considered. Okay, I'll take the next slide. So one of the key things we want to highlight in this study is that this study, or developing a risk profile, while we might have a universal scientific opinion, we have to be clear that this is highly dependent on the company in question, you, the client, the manufacturer, what business you're in, and it's also what your own risk appetite is. So I want to make it clear that what you're seeing here is just an example, okay, that would have to be fine-tuned for different manufacturers. So just starting on the right, when we say very high risk and we say it's red, that is where it is very clear in the regulations that you are not allowed to share these modalities with any other modalities. So penicillins, beta-lactams, these are highly sensitizing chemical agents that regulators just simply expect to be completely separate to anything else, okay? If I jump onto the left, we're now looking at some manufacturing processes that have very little chemical, sorry, not little, but lower chemical hazard. So in terms of, and we're looking at just potency and toxicity here. So take your traditional biologics MAB facility where you're dealing with caustics, acids, that could be quite common across many modalities and are used regularly in the biotech industry. So we look at our mammalian cell culture facilities, and we look at our aseptic fill finish, and we have CMOs who are running a buffer and media prep business where they're selling bags of buffer and media. So these are kind of examples of how a manufacturing space would have a low potential potency or toxicity. One of the things we need to flag here, there will always be exceptions. So some monoclonal antibody processes use methotrexate, for example, in their inoculum lab. So again, I want to highlight here that this is very company specific, and there might be some exceptions to what's happening in different risk groups. So they're the two extremes. We got the high potency and we got the low potency. And then you have to start working what's in the middle. You know, so one good way of drawing a scale on potency and toxicity is to look at the OEB band. So our low is less than OEB 3. If we're getting kind of close to being equal to OEB 3, and we start hitting medium risk, and then once we go beyond OEB 3 into OEB 4 and 5, we're now in high risk. So just to put some examples there, you know, from a medium risk, maybe filling the vials of an ADC process. So we're not handling directly the payload of an ADC process, but once it's now conjugated to the monoclonal antibody, solubilized, there may be occupational hygiene data to say that, yes, there's a toxicity risk, but it's not as high as maybe the actual ADC process itself. So then next risk profile is to turn around and say that we are conjugating the ADC now. So now we're actually handling the payload, we're handling high potency anti-cancer target molecules. We might be making some peptide molecules, for example, where the OEB number, OEL exposure limit starts to rise, and we now need to start worrying about that chemical molecule cross-contaminating other manufacturing suites. Okay, so again, I want to highlight here as a key point This can be very specific to different manufacturers. They might have their own risk appetite. They might put certain things in medium risk that other companies might have high risk, etc., etc. The key thing I want to highlight here is red is where the regulators very prescriptively say that must be separate. Okay, Alf, give me one more slide. So when assessing multimodality for potency materials, high potency, low potency, the ISPE already has great publications to help with this approach. So we have here the risk map methodology that they use. So this is where they establish, you know, that there's 4 key risks that you need to assess. And we're not going to spend too much time on this today, but you have mix-up. So did I deliver material to the wrong manufacturing suite, for example? So now a high potency material is accidentally in a low potency suite. Have I any carryover from process equipment? So do I have stainless steel equipment that is cleaned and now a CIP skid that's shared has now been contaminated? Maybe I'm single use and that carryover doesn't happen because all my components are removed after manufacturing. And mechanical transfers, that is transfer from an operator. So again, it has a methodology that if my suites are sharing a locker room, for example, do I have a risk that operators from different suites are actually able to transfer material onto each other? And then airborne is through our air handling system. So the key thing I want to highlight here is that there's loads of really good material by the ISPE to help with this. Okay, I'll take one more slide. You know, so from a regulatory perspective, some things are clear cut, black and white. There's an area of gray for kind of OEB4, maybe some OEB5 material that we have to assess on a case-by-case basis. And then there's some stuff that are relatively low risk, okay? And there's good data and publications already available from the ISPE. I'll go again off. So now we need to start looking at that there's no chemical hazard as such in my manufacturing processes or in my multimodality facility, but there is differing and escalating either viral or microbial risks. So let's do the same again. So on the right, we have BSL-3 and BSL-4 organisms. These are highly harmful organisms. So again, these are called out specifically in GMPs that there's an expectation that these organisms are in separate manufacturing facilities. So on the other side, we have low risk activity. So anything that is BSL-1. Okay, so this is the lowest biological hazard. Okay, so here we have typical examples might be your CHO cell line for monoclonal antibody manufacturing. So there's a few things here to highlight. A, the CHO cell itself is very low risk, but secondly, any virus that might propagate in it is not likely to be a human virus. It is probably some sort of murine mouse virus. If I move into the next risk category, so anything that might be BSL-2, so we now worry about yeast, E. coli manufacturing, any fermentation processes. But also we have viral vector processes, and these are processes where I have human cell lines. So I might have HEC273, which is being used to propagate a viral vector. Now, that is a human cell line. A human virus like influenza or COVID could potentially propagate in the bioreactor. But the key thing here is that is a characterized cell line. So you're, as a manufacturer, you are ensuring that the cell line, when it's used at the bioreactor, is in principle, you have screened it to make sure it is free of most human viruses. So we're saying, like, in theory, there's no human viruses present, but it's possible that a human virus could be present, okay? And then our viral vector process, again, the vector in theory should not replicate through the human. So through a human, as in an operator in the manufacturing facility, etc. It can only replicate in the human cell line. One of the things I want to highlight here, again, just to reinforce the point that these risk profiles are relative and they need to be fine-tuned to different manufacturers. So you get in the world of gene therapy and viral vectors, there are Sometimes there are human cell lines and the viral vector, but some processes have a wild type helper virus in the bioreactor as well to help with the propagation. So you might have to turn around and say, well, actually, no, there is real human viruses in the bioreactor in that gene therapy or viral vector process. So again, we strongly highlight that, you need to case by case this with your manufacturer. And then we get to the high risk biologics processes. So this comes to the question that Alf had asked in the poll at the start. So if I have live virus manufacturing, so I actually am culturing a real virus in a bioreactor to make a vaccine, autologous cell therapies, this comes up quite a lot. So you might have, if you take all the approved CAR T cell therapies at the moment, they are real patient cells. Those patient cells have come from a hospital. Those patients are immunocompromised because they've had several rounds of chemotherapy. So those manufacturing facilities assume that real viruses are present in those patient bags. So an autologous cell therapy facility is working on the assumption that real viruses are in the manufacturing process for all these individual patient batches. So in the polling questions that we asked at the start of the presentation, this is why this is the highest risk. An autologous cell therapy, you are knowingly bringing human viruses into your manufacturing facility, okay? The reason a monoclonal antibody was the lowest risk is because you are screening it for viruses. It's not a human cell line, so a human virus in theory shouldn't replicate through a bioreactor, and that's why that would take the lower risk, okay? And again, with all risk, there's no risk, it's just low, medium, or high, okay? I'll take another slide off. So just that's a summary of what we said. I'll keep moving on for time to give people a chance for question. So one of the things I want to highlight here is that multi-product or dealing with different chemical entities in a manufacturing facility has been around a long time in chemical manufacturing. That's why the ISPE has the publication on risk map for the toxicity and potency side of our discussion today. There is no document yet about how to assess risk in terms of viral vectors, autologous, real viruses present, bacteria. So the point is we do have some guidance. We do have ISPE baseline guides for monoclonal antibodies. We do have the latest autologous cell therapy guide from the ISPE, but we don't have an equivalent to risk map for virus and microbial cross-contamination risks. Okay, Alf, I'll take another slide. Okay, so something actually I forgot to mention actually, there is a methodology in risk map for the potency side, where if you do have two chemical entities in one suite and a different entity in a different suite, where there's a concept called the permitted daily exposure. And these are an analytical way for you to be able to test that how much actual of chemical A needs to get into product B to actually cause a cross-contamination event. That informs your QC release and swab testing, that you're able to verify that you do not have cross-contamination between your manufacturing suites. There isn't an equivalent concept in virus or viral load. It's just generally assumed by manufacturers that you just should have no virus from process A getting into process B. But one of the things that we highlight with a lot of our clients is that it's very useful to have data that if you do have mixed modality manufacturing and you do know that certain vectors or viruses are being handled in different suites, that you have robust data on what your biokill systems do, what your VHB decontamination does, what your spore cleansing, your routine cleaning, that you have data to say what sort of log reductions you can expect from your cleaning regimes, from your sanitizations, sterilizations, whatever, okay? I'll keep going. And again, just as a general point, as you work up through the BSLs, you can take that as a rule of thumb. The more higher BSL you have, the bigger thing you need to worry about. We'd also just flag, it's that idea of viruses. Are there, only murine viruses potentially present for MAB? maybe there's only viral vectors present for our medium risk, but then high risk is there's real human viruses present. Okay, I'll keep going off. Okay, so now we develop a risk matrix. Okay, so this is where, you know, as a company, as a manufacturer, and you want to understand what you do, you need to build a profile of how you as an organization has categorized what modalities are in what zone. Just one thing that we typically don't see a high risk in both microbial and potency. We tend to, it's either one or the other. It's either a high potency risk or it's a high viral risk. But we never see that it's both high potency and viral and microbial. Just there's a few things here, like, so, you know, most companies, you know, the reds are outside of the area that you'd consider in the study. You know, you might consider that, you know, what are, you know, therapeutic proteins, vial filling of an ADC, or what we have at the top there, the antibody drug conjugate. You might consider that. We would have some companies, so particularly on the virus side, and just to give you an example, we have one of the main CAR T players at the moment. They would simply say that is a separate business completely, 100%, and I will never ever share a mixed modality platform with any of our other products. And that is purely because as a corporate entity, you know, CAR T is a separate business, completely separate site, and that might not be for any theoretical risk. It's just purely that's how their company is set up. So these risk profiles that we set up, they're highly specific to you, the manufacturer. They're highly specific to your perception of risk, and they're highly specific to your business. Okay, Alf. So as we highlight here, generally when we do these studies, the first step is to build this risk profile as a theoretical exercise. Okay, Alf's going to talk through some examples, you know, but at least we develop a starting point of what your perception is of risk for all the modalities you're interested in. And we agree common understanding of what should not be accepted based on regulations, but we can't stress enough that, you know, this is only a starting point graph. The next point that's going to be very important is that we do the full mixed modality assessment. We do some risk assessment, which Alf is going to talk through here. And then it's only at that point that we will finalize what your company's profile of what modalities are acceptable to be in the same manufacturing and what are the rules that are required for putting those two modalities beside each other.
All right, thanks, Tom. So having gone through all of that, then let's have a look at facility features by risk group. So having developed the risk matrix and the risk profile, the next step is to then develop the various facility level features by each of the different risk groups. So, for example, this might be, is it possible to share common areas and support services such as corridors, supply corridor? Do we need a return corridor? Can we share locker rooms? Can we share HVACs, utilities, wash areas, clean utilities? So that's what we mean by facility features by risk group. And then once we've established all of that, the next step then is to then start performing the risk assessments and to look at our assumptions and just satisfy ourselves that what we've developed is going to work, and we're not going to have any risks that are going to cause us problems in relation to having multiple modalities within the same facility. Okay, and as with all risk assessments, they should be performed with a cross-functional team and making sure that we have the right level of segregation. So we have a diagram here showing to the left the different risk categories, whether it's potency related or whether it's to do with the microbial viral contamination. So you'll recognize the risk groups there, and at the bottom here we have the very high chemical biological risks, which we're saying are out of scope and will require dedicated facilities based primarily on the regulations that we covered earlier. So if we look at the modalities that are in scope, we would look at the architectural aspects of the facility. So that could be the corridors we mentioned earlier, whether we'd need a dedicated waste corridor, for example, whether we need a return corridor as well as a supply corridor. So, we would have unidirectional flow. In terms of the HVAC, as I said earlier, can we share the HVAC? Do we require segregated HVACs? What would we require in terms of pressure cascades and other factors? When it comes to support areas, it might be that we might have a wash area. Again, does that need to be dedicated? Can we share a wash area? Can we share autoclaves? And also the warehouse. What aspects of the warehouse can be shared? What might need to be dedicated? And then finally, utilities. So, this could be purified water, Wi-Fi systems. Is it okay to share those systems, or do we require dedicated utilities? So once we've developed that, we then perform the risk assessments. And the way we do that is by identifying the potential failure modes, and we assess those. And then so we perform, and often the way we would do that would be through the FMEA process. And I've mentioned here ICH Q9 as well. So there is a new version of ICH Q9 that's coming into effect this year. And for those of you who haven't already read ICH Q9, I would recommend that you have a look through that, because it does contain some useful information that's particularly relevant for these risk assessments. Okay, so finishing off then with a case study. So this is a study that Tom and I worked on a number of years ago. It's from a CMO whose primary business focus is on biological products. And so this particular CMO had constructed a very, very large agnostic facility, and they'd already invested in a lot of the infrastructure, for example, with the clean utilities. It consisted of a number of different floors. On the ground floor, it contained a JIT warehouse and various support areas, including waste management. Material is supplied to each floor via an elevator from the ground floor. And there are additional elevators as well that can be used or be made dedicated for waste streams. And the idea or the concept was that the facility would accommodate multiple modalities, including some of the floors themselves. Each floor might potentially accommodate multiple modalities. And the CMO was interested in understanding, which modalities might not be suitable for this facility, and of the modalities that they were interested in, how they might accommodate those modalities. So that's where we came in. And this is what they concluded. So for low-risk modalities, it was decided that low-risk modalities could be accommodated on the same manufacturing floor with common approaches to flows, gowning, airflows. They would be able to share a loop corridor, which you can hopefully see here to the right, as waste material can be easily contained prior to leaving the manufacturing space. However, it was decided that within the manufacturing space there should still be unidirectional flow, and that material and waste flow should have their own dedicated lifts, which was possible. You can see the waste elevator lift to the bottom left here, and the separate supply elevator here for materials. So those are low-risk modalities. So for medium-risk modalities, so for example, viral vectors, viral antigen vaccines, and allogeneic cell therapy, it was decided that a separate waste corridor would be needed due to the risk of cross-contaminating viral material. Similar to the low-risk modalities, we would have badge access control to control personnel movements. So, for example, we have an example here. So personnel accessing a manufacturing area would not be permitted to enter another space without first returning to the locker room. And we would have swipe access control. So to the diagram here, you can see the different manufacturing spaces. You can see separate supply and waste corridor. and the same configuration with the waste and supplier elevators. Okay, then finally moving on to high-risk modalities, it was decided that these modalities would need to be isolated and should not share locker rooms. So it's felt that one manufacturing process should take the entire floor and not have multiple high modality processes adjacent to each other. It was decided that the risk was too high. And if, for example, we're looking at an autologous cell therapy such as CAR T, and if the floor was too large for the type of modality that is being considered for the facility, then it might be that a dedicated facility would need to be built specifically for that modality. Okay, so if we were to conclude then, the outline approach begins with identifying the guiding principles. It involves working within the regulations, and it involves developing a basic risk profile to determine how best to accommodate the different modalities, bearing in mind that incorporating the different facility constraints that exist. And as Tom, I think, explained, that will be different from one company to the next based on their risk appetite, based on the operational constraints that they may have. So whilst each facility may be different with a different set of recommendations, we do think that the approach that we've gone through and the methodology can still be applied, and not least during early feasibility. This approach is designed to help understand what modalities may be suitable for a given facility and how modalities may be accommodated. The other point that I think is worth making here as well is that normally when we design facilities, we might make a number of assumptions during the feasibility study. And as we progress during the design, we may learn more information about a particular product or a process. And we'll definitely have more information about the facility, the equipment. What we need to do during each of those stages of design is to circle back and then just challenge some of the assumptions that were made during the feasibility and just verify that they still hold true, because it might be that as more information becomes available, we may need to reassess some of the earlier assumptions. We may need to revisit some of those risk assessments, and we may even recategorize a risk category for a particular modality. And I think that concludes the presentation.
Just something. Can you go back up to the medium risk scenario there, just to?
Sure.
I'll add a couple of points. So this is the crux of the whole methodology. So the first part is we did a theoretical exercise where we identified the modalities and we categorized their risk. And then we did a second theoretical exercise where we associated medium risk with certain architectural facility features. So here we said that medium risk stuff need a dedicated waste corridor. And we might have said, and we just showed it for brevity here, that we might have said that they need segregated HVAC. We might have said that manufacturing operators need to be dedicated to suites and they're not allowed into other suites, for example. So the key point I want to highlight is that there was two theoretical steps: identify the modalities and their risk, and identify the features we think should be in place for those modalities. The third step is that we did some, not made up, but we did some risk assessments where we actually got a cross-functional team to now start challenging, for example, the supply corridor here. What if two operators bumped into each other from the two suites? Did they transfer potency material from one operator to the other? Did they transfer? And we go through that FMEA, because then what happens is that once we complete the FMEAs, we update the theoretical risk matrix and the facility matrix to say this is the final playbook for what we think is acceptable in your company's context and for mixed modality manufacturing. So that's the key thing. There's two theoretical steps. There's a risk assessment step, and then we update the two theoretical steps. And now you know what's acceptable to manufacture what beside each other. So we've been quite simplistic on some. So for example, here on these slides, I've just walked through the major corridors and flows of personnel, but there's obviously HVAC equivalent to these slides and separate HVAC, etc. But we just want to give you a flavor of the methodology. Sorry, Alf.
No, that's good. So I think at this point we can probably open up to questions if there are any.
Thank you, Alf. Yeah, thank you, Tom. So would anyone like to ask a question, or you can please do post that in the chat if that's more convenient. And I'd like to thank Alf and Tom for plugging the ISPE publications as well. And obviously that's a great resource for everyone to go to. This session is actually being recorded so that you can actually refer back to it on our ISPE Singapore YouTube page. So if you missed any of it or you would like to watch a rerun, please feel free to do that. So I'd like to thank Shan Shan for her question. She's just posted, can you please elaborate the point that dedicated autoclave required for medium biological risk?
So go back to the I think did we say we could share an autoclave?
This is for, sorry, which category?
Go to the facility matrix corridor. And I think we said there that it's
this one here, I think, isn't it? We're talking about.
Shan Shan, is that correct?
So the reason we're saying that is that if you have, let's say, 2 or 3 manufacturing facilities are sharing a washroom, people start getting concerned about manual cleaning of parts and cross-contaminating them. Yes, you probably could justify that an autoclave, as long as it was passing through into a clean area, that you could share the autoclave. But again, I suppose the point you have is that a lot of manufacturers would just get concerned because, if I'm disassembling stainless steel parts, bringing them to a common clean room, they might start to get worried of cross-contamination of potency material or viral material or microbial risks. That's all.
I think, Tom, it might be worth talking about the waste as well, because I know sort of medium, high-risk facilities, there are some organizations that will insist that all the waste is decontaminated in an autoclave before it leaves the production area. Others are, happier for the waste to then be collected and maybe taken off site and incinerated or whatever. So there is that aspect as well, isn't there, that needs to be taken into consideration.
Okay, thank you both.
Yeah.
Any further comments, Tom?
No, so do you want to go through, I think Gary has a question. How much?
Yes.
Okay.
Yeah, so Gary asks, how much is the consideration of volume handling in the cross-contamination between products and suites? So
it's a very good point. So there's a certain, in mixed modality manufacturing, generally, if you have process A is a 50-liter scale single-use facility, and then process B is 15K 6-pack bioreactors. There are certain things that, it's just the sheer scale of the process means they'll never be beside each other, just because it's just the limitations on your building from a flexible point of view. But you're right, when you get into your risk assessments, if you have a lot of solid waste coming out of a corridor area, and you need to quantify the nature of that waste. Is it, kind of, let's say, wet depth filters, for example, that aren't sealed away in bags? They're all classics that people would have to bring up to say that there's more credible risk of dripping waste in the corridor, etc. So yes, I think when you get into your risk assessments and you list the manufacturer's modalities, you have to start digging into credibly, is it a high solid waste manufacturing process, or high volumes of product going to the warehouse or materials, because that all comes into the, so for example, if a defense is time-based segregation, if the volumes are huge from one manufacturing suite, that defense starts to get harder and harder, and that should come out in your risk assessments as a challenge.
Thanks for that question, Gary. And JP posts a question to which Gary also had a follow-up. The question was, for high risks such as autologous cell therapy, how is cross-contamination between batches contained? And Gary has a comment, this isn't about potential viral transfer, but rather that patients don't get back their cells, which is a different type of risk.
Yeah,
Comments?
so I think, yeah, so maybe JP's question is a risk within the manufacturing suite, okay, of mix up of patient batch. So just to answer that question, a lot of it comes down to in cell therapy workstation design, that any patient batch is in a dedicated workstation where no other patient batches are allowed in it. There's all sorts of technologies with RFID tags that, you know, the operator bag, so the patient bag is booked to a workstation, and the barcoding system makes sure there's no other patient in that workstation, etc. So there's a mixture of architectural design that prevents human nature from mixing, but there's also digital technology around barcoding, scanning, RFID, proximity RFID, that the list is endless. So that risk within the suite is somewhat separate. In a mixed modality context, is that when I then step out of that suite to share a corridor, we would say that you have to recognize that that patient material is a real human with potentially real viruses in it. So that's where you start to worry about other suites. Or I think to Gary's point, I might have a viral vector manufacturing next door, and I don't want to inadvertently hit the patient batch with the wrong viral vector. So that's where the you know, on a mixed modality context, you would assess those risks from a cross-contamination point of view. I hope that's answered your question, JP.
Thanks, Tom. Does anyone else have any questions? This is still your last chance to get Alf and Tom live.
Am I allowed to ask a question, Linda?
Yeah, of course.
A bit unusual, I know. During the poll, the second question was you know, which one was the highest risk modality? And the majority of people thought the viral vector was a higher modality than the autologous cell therapy. And I just want to make sure that, you know, I just want to understand if we've been able to answer that question as to why autologous is considered to be a higher risk or not.
Okay, good question. Would anyone like to comment from the audience, please? No news is good news.
Okay, that's good. That's all right.
But thank you for rechecking that out. Okay, so JP has a question.
JP followed up a great point. So one of the key things in autologous cell therapy is that it all comes down to the approval. You know, so yes, you know, there is, all the CAR T companies have multiple patients in the one room, and they are working on the assumption that a different patient could have a virus and a different patient. So it's a brilliant question, and it comes up a lot in cell therapy world. The key thing that you need to understand is that a lot of the CAR T therapies are approved as the last medication for a lot of these patients. So regulators inherently understand the risks of CAR T cell therapy. And you'll see, if you read any of them, you'll see that they have to have had 2, maybe 3 attempts to cure their cancer with other medication before they resort. So it's inherently understood by the manufacturer and regulators that CAR T, for example, has some higher risks in it, but it's balancing that against the patient has no other medical options available to them.
And I think Gary has pipped in on that one as well, in terms of stating that autologous patients are also tested for viral contamination, but you can't check for all virus in the world. So yes, autologous patients are higher risks, and some have other viruses, e.g. plasma potentially as well.
So one thing there, like, so it's a brilliant debate. Now I think a lot of these questions are what's the risk within a CAR T suite as opposed to mixed modality, but European blood directives, diseases like HIV, Hep A, Hep B, they all have to be screened in blood transfusion world. So in a European facility, CAR T facility, I don't know about Singapore, but a lot of other countries, that by law, blood banks have to screen for a selection of diseases, while in America, they're not mandated to do that, in the US. So probably to where Gary's going, in some cell therapy jurisdictions, there is screening of some of the major diseases, but in others there isn't. So again, the whole point of, yes, there is a potential that a virus will jump. And this comes down to closed processing and segregation of workstations, etc. The risk is still relatively low, or can be made as low as possible, but it's all because that patient has no other medical options available to them. And that's the context that a lot of those therapies are approved in.
Thanks for making the distinction between regions. And JP asks, if we assume each cell therapy patient has a human virus, why are they all combined in a single ballroom?
So again, it's just risk. So yes, at the very start of cell therapy, 5, 6, 7 years ago, there was some companies doing one patient, one room. And the problem is the buildings get massively expensive, because while the more you aggregate or combine into, now when I say ballroom, it might only be a 100 square meter room and there's max 10 patients in the room, but the more you allow multi-patient in a room, the more, I suppose, cost effective the building design is. But you're asking great questions. That all has to be balanced against, can you guarantee no aerosol? Can you guarantee your closed processing does stay closed? And if you do have to do open processing, can you guarantee that your workstations are rigidly segregated and there's physical screens separating one biosafety cabinet area from another biosafety, so that the risk is as low as it can be, that I don't get inter-patient batch cross-contamination.
Thank you very much, Tom. Does anyone else have any other questions? We've got 5 minutes remaining of this session.
Yeah, hi, Linda. This is Harsh from Intel.
Hi, Harsh.
I'm curious, if you can speak on automation. How does it help in lowering some of the high risks, areas, and anything about automation? It is a very general and vague question, but I want to hear from you.
So the more automated your process is, the likelihood is the more closed your process is. And process closure is fundamental to everything. So if you take two suites beside each other and they were both highly manual open processes, for example, you now really run the risk, and your risk assessments should be highlighting that people mixing in corridors is not acceptable. Or, you might, while we've shown an example of medium risk modalities, can share the people corridor, but ways to separate. If everything is manual, you might stop and say, we need to readjust our facility matrix, because the risk of people mixing now is much higher, because I got two different modalities, both are open and both are manual processing. Does that make sense? While if somebody says no, strictly, they are closed processes, therefore the presence of viruses or whatever the cross-contamination concern is, that the presence of them outside of the bioreactor or reactor becomes a hell of a lot lower. Okay, so the point really is that the more closed, hence automated systems that you have, where you have an automated pressure test, you have an automated CIP, you have an automated etc., that all is a massive game changer in how you manage risk. Does that make sense?
Thank you, Tom. Yeah, it does.
Thanks for the question, Harsh. And we have another one, which is between different batches, is it suggested to always VHP versus other methods, even in low-risk modalities?
Will I take that, Alf, or do you want to take it?
Yes, I would say that would have to VHP all the time. I think if you're handling a viral vector, it might be that in an isolate, for example, you're more likely to want to VHP that, but you wouldn't necessarily want to VHP a whole room. So I think it depends on the product, the process, and I think there are probably many applications where they may not be necessary to VHP. Tom, do you want to?
Yeah, so this is brilliant. It comes down to our risk matrix. So we've got our low, medium, and high. So this is the challenging part of it, is that and I'll go back to the high risk, slide 39, Alf.
Yeah.
Just so if I take low, medium, and high risk, so like if I say a low risk modality, your normal floor, ceilings, and walls and ceilings cleaning regime with your Vespine, your Spore Cleanse, whatever chemicals you're using, that is probably appropriate for low risk manufacturing, the risk of something. But then if you go to high risk manufacturing, if I had a room and I was going to do a couple of batches of product A and into product B, yes, the manufacturer may choose to have a room. So we see that sometimes in viral vector CMOs, that their upstream and downstream areas are now going to transition to viral vector B versus viral vector A, and they will VHB their room. But we are seeing that in the middle risk or the medium risk zone, it could go either way. A CMO who is, wants to reassure their customers might choose to VHB, while a in-house company who's making their own viral vector for their own cell therapies, they might be happy that they can do a cleaning regime by just traditional, mopping floor, ceilings, and walls approach. So it depends, but I would say for low risk modalities, we generally wouldn't see, from a cross contamination point of view, we would not see VHP. But yes, naturally, an aseptic filling line will VHP purely because of trying to keep the environment decontaminated and for aseptic processing. I hope that helps.
And Tom, we know as well that VHP can impact the efficacy of a product as well. So there are cases where maybe 30% of the efficacy of a product has been impacted by introducing VHP. So that needs to be considered and mitigated as well.
So these are all brilliant questions. So why would I VHP the corridor? If your risk assessment said that there was genuine risk in the corridor because of mechanical transfer of viral material from one operator to the other. Now, again, the reason I got Alf to go back to this here is that this was a company who they were not, if something was high risk from a viral perspective, a live vaccine or something, they just didn't think they could defend how clean the supply corridor would be, for example, or a shared corridor. So they were saying, look, and this probably going back to your point that maybe they were thinking they would have to VHB corridors, and then maybe they thought, and they just went, no, we're done. We're not sharing a high risk with another high risk. It's a dedicated facility with its own locker rooms. So we don't need to do that kind of cleaning of corridors. So those ideas would all feed into your risk assessments to see what is the risk on a shared corridor. Could you have a mix of viral material and so great questions, but they would all feed into your risk assessment.
Thanks, Tom, Alf, and great questions, everyone, and great answers. I hope everything was covered. We have now reached the time for the end of the session, unless anyone has one final burning question to ask. Otherwise, we will wrap up for this evening. And I'd like to thank Alf and Tom for sharing their expertise and time with us this morning, your time, and to let you get on with the rest of your day while we end ours in Asia. Thank everyone for joining us this evening, and we're looking forward to seeing you all back at future sessions. So thanks again all, and look forward to seeing you.
Thanks, everybody.
Thank you.
Bye.
Thanks,
Thank you.
everybody.
Bye-bye.
Bye.